Dear students, in this lecture we are going to know how the cell biology developed and will study the history of cell biology. It began its journey in 1665 when Robert Hooke discovered the cell. Actually, he discovered the cell wall of the dead cork cells and he named them cell. In 1745, Bonnet he discovered natural parthenogenesis, and most of you must be knowing that parthenogenesis is the development of egg without fertilization. In 1772, Priestley and Ingenhousz said that carbon dioxide along with water in presence of sunlight produces oxygen, and this phenomena takes place in plants during photosynthesis. 1784, Fontana discovered nucleus in the skin of eel, a fish. In 1824, Prevost and Dumas described cell division for the first time in the cleavage of frog's egg. It was for the first time that it was stated that the cell it divides. In 1827, von Baer discovered mammalian ovum. Then in 1831, Robert Brown discovered nucleus, which is centrally placed mostly in most of the cells. 1839, M.J. Sleeden and T. Swan, they put forth cell theory. Then in 1839, Purkinje and Volmont named the living material of the cell as protoplasm. Rimac described amitosis, which is the process of cell division in RBC of chick embryo. Then in 1854, Rudolf Virchow completed the cell theory by stating that new cell arises from pre-existing cells. In 1870, Friedrich Miescher isolated for the first time nucleic acid from the pus cells. 1873, Snyder described chromosome for the first time. In 1875, Van Benden observed centriole. O. Hertwig in 1875 described fertilization for the first time, and he said that fusion of two cells from or gametes from different parents is called as fertilization. 1876, Louis Pasteur described anaerobic release of energy from cells by fermentation, and he had seen this process in yeast and molds. 1879, W. Fleming described chromatin. Chromatin is DNA associated with proteins. 1881, Balbiani discovered giant salivary chromosomes, which you called as polytene and Lambert's chromosomes. In 1882, W. Fleming for the first time gave an accurate mention, an account of chromosomes, and he also gave the term mitosis. In 1882, Fitzgerald discovered chromomeres, which are beaded appearances on the chromosomal length. Machman described cytochromes for the first time in 1886. In 1887, von Benden said number of chromosome reduced to half during gamete formation, and this was the reason for the constancy of the chromosome even after fertilization. This was very important discovery. In 1897, Altman and Benda observed mitochondrion which were the seat of energy generation. 1898, Benda named mitochondria as mitochondria. Palade in 1952 described cristae which are present in mitochondria, and Camillo Golgi in 1889 discovered another very important organelle in the nerve cell of Barnwell by silver impregnation into the cell. In 1900, J. Loeb able to perform artificial parthenogenesis. 1901, Montgomery said that they are the only homologous chromosome which undergoes synapses in reduction division, not whole. Michaelis and Menten in 1913 mentioned that enzyme action, enzyme and substrate together forms enzyme substrate complex, and after enzyme substrate complex the enzyme act upon the substrate and the product is released and formed. In 1914, Lilly gave fertilizing theory. It mentioned that egg produce fertilization and sperm produce anti-fertilization, and it is because of the action of and reaction of anti-fertilizing and fertilizing, the sperms and ova clump together and fertilization is realized. In 1926, O. Warburg discovered cytochrome oxidase, which is related to catalytic oxidation of oxygen, cytochromes by oxygen. Then 1929, Lohmann discovered a very important molecule which is adenosine triphosphate. In 1931, Lewis discovered pinocytosis, which is cell drinking. In 1934, Bensley and Hoare isolated mitochondria from the live cell. Stanley isolated tobacco mosaic virus in crystalline form and could able to demonstrate that viruses are non-living. 1937, Krebs discovered Krebs cycle, which is also called as citric acid cycle. In 1941, Claude isolated mitochondria by ultracentrifugation. 1945, Porter discovered endoplasmic reticulum, a very important organelle of the cell. Lippmann in 1945 discovered coenzyme A. In 1952, D. Dove discovered lysosomes. In 1956, Sparks and Pons discovered quantasomes, which are present in chloroplast. 1963, Chance, Parson and Fernandez Moran discovered elementary particles in mitochondrion. As far as cytogenetics is concerned, Kohlreuter discovered quantitative inheritance, that intermediate characters are observed in hybrids. Gregor Mendel, famous laws of heredity were given by him in 1866, and in 1833 Raux stated the role of chromosome in heredity. Hartwig and Strasburger stated the role of nucleus in heredity in 1883. In 1900, Hugo de Vries gave very important mutation theory, observed certain characters in O. Lamarckiana. McClung in 1902 discovered sex chromosomes for the first time in grasshoppers. 1906, Bettison and Punnett discovered linkage of hereditary units. Some traits remain associated for generations. This is called as linkage. In 1909, Jensen's discovered chiasmata, that the exchange of chromosomes segment takes place only because of the formation of chiasmata during the process of crossing over. 1910, Morgan put forth gene theory and sex linkage in Drosophila. Shull, 1914, gave concept of heterosis. and stated that the hybrids have greater vitality and vigorosity than their parents. C.B. Bridges stated that non-disjunction in chromosomes of Drosophila and the both pairs which remained uh, associated passed to same parent. Winkler concept of heteroploidy. C.B. Bridges' genic balance theory stated that sex in Drosophila is determined as a result of balance between autosomes and X chromosomes. In 1927, Muller artificially induced mutation in Drosophila by X-ray irradiation. Griffith in 1928 demonstrated transduction in bacteria. McClintock 1931 explained crossing over phenomena. Beadle Totem induced biochemical mutation in bread mold, which is called as neurospora, by X-ray. Sonborn extranuclear inheritance in paramecium was demonstrated by Sonborn and explained in 1943. 1944, McCarthy and MacLeod transformation in bacteria. The famous transformation in bacteria was demonstrated by McCarthy and MacLeod, in which the virulent form, non-virulent form became virulent form. In 1945, Barr and Bertram explained the formation of Barr body and explained what Barr body is. It is heterochromatization. 1956, Zhou and Levin first correct count of human chromosomes was given by them. So thank you very much, students. This is all in this lecture.